I'm your host, Jeff Wu.
And I'm my co-host, Michael Brandt.
And we're stoked to have Megan Klimen, founder of 3Scan, with us today. And was recently profiled, part of New York Magazine's article on biohackers and being one of the more prominent female biohackers out there. So we're excited to have her on board on this episode to talk more about her biohacking experience and also about her entrepreneurial journey as not only a founder, but also a founder in a space that's just burgeoning.
Awesome, and I'm super excited to be here. Like I was telling you guys before we started recording, I'm a huge fan of what you're doing, and I really think that you guys are just pushing forward exactly what we need to be doing in biology, like exactly what we need to be doing as people to always be thinking about how to improve ourselves, how to improve our cognition, how to improve, our basic biology.
Awesome. Yeah, I think that when people think about biology, it's always stereotypically been one of the more fuzzier sciences compared to like your pure mathematics, your physics or chemistry. Biology has always been more of an observational science. What do you think have been the biggest changes have moved or developments that you've seen over the past years, decades, that have moved biology more and more quantitative?
Yeah, I mean, I think that just that acceptance that biology should be something that is quantitative, especially when you're talking about moving things into the clinical world and clinical diagnostics. Like there's just really there's a lot of slowness. Like people really hesitate to change the way that we do anything in the world of diagnosing diseases. And pathologists are kind of the worst at this. Looking at tissue and determining a disease from the visual inspection of tissue has been so much more of an art than a science.
Right.
Since its inception. Very little has changed in the way that we look at tissue and the way we do the basics of histology and pathology. And it's been really great in the last few years just to see this idea of, things needing to be more quantitative really take hold. Like as we've been able to see advances in computing, it's made it feasible to look at visual data from tissue and have computers be involved in diagnoses.
Do you think that this old sense of biology is going to go away and that biology is just going to become computational biology?
Yeah, absolutely. I mean, I think that we do things to the best of our ability at the time that we're doing them.
So it's like a tooling thing. We just better animals with better tools now. Is that
exactly? And I think that the tissue scale biology has been one of those holdouts, right? Partially because we didn't have the tools, we weren't able to. It's still really hard for computers to look at pictures and be able to pull out relevant information from it, right? But part of it is when you don't conceptualize that as being a problem, you don't work on fixing it.
Yeah, I think that's a funny analogy where the car used to be called an automatic horse carriage, and now it's just an automobile, right? And I think the same thing will happen with biology. Now, I think that dovetails very nicely with talking about computational, vision applied to biology. To talk about 3Scan. I mean, you guys just recently closed a big series of funding. I think our listeners would love to hear more about 3Scan, the technology you guys are building, and a little bit about your founding story there.
Yeah, absolutely. So 3Scan is a microscopy company. We run imaging as a service. So people send us tissue, we image the tissue, and then we perform some degree of analysis on the data stack that we have from it. What makes 3Scan really different is that we automate the process that is traditionally really manual. So if you want to look at a piece of tissue, and it doesn't matter if you're a researcher doing this for basic drug discovery or a doctor pulling out a biopsy to make a diagnosis, the way that you look at this tissue is pretty much the same and has remained unchanged in the last 150 years. You take that piece of tissue, it's really squishy, you can't cut a thin section from it, so you put it into a hard substrate. Usually that's a paraffin wax if you're doing light microscopy. If you're doing electron microscopy, it's going to be a hard resin. You then cut a very thin section. Usually that section is 1 cell thick, about 5 microns. You use that thin section, you cut it off on a thing called
What's the technique to cut such a
So you use a thing called a microtome, which is kind of like a deli slicer. So it's like
That's
a deli
one
slicer
cell
if you're
thick.
doing
Right.
like nano bacon, right? Like so you use this microtome, you cut off a thin section as a human doing it. You then float that section on a water bath. You pick it up on a slide. Then you take all the steps that you did to put that tissue into the wax. You take all the wax back out. And so you put a solvent on it, you put you put alcohol on it, you put water on it, and then you stain it, and then you look at it.
So in the clinic, are we, putting the deli slicer onto your arm and taking a slice of bacon off of my skin?
So the deli slicer is the traditional method. So after that doctor comes in and takes the biopsy out of you, once it has walked away, then it's gotten deli sliced.
Okay.
The problem with doing that all by hand is that's incredibly manual and incredibly slow. This is why we take a sampling, right? This is why it only makes sense when we're looking at a piece of tissue to take, 3, maybe even 20 sections, but use that as a representative sample and hope that we're going to see whatever characteristics we need to see in there. So let's say, you've got a weird looking mole on your arm, we pull that mole out. I'm going to go into my lab. That's, that process is going to go through, like a histotech's going to take, your sample, take it through, put it into a block of paraffin, take that thin section. They'll take a handful, probably 5 representative sections. They'll stain them, and then the pathologist will look at it and tell you, hey, is your mole healthy, or do you have cancer? Now, taking 5 sections from a thin biopsy that was from your larger mole, that's a sample of a sample of a sample, and we hope that we saw the cell that is going to give you the right diagnosis. But the chance that cell, that abnormal cell that's going to give you the right diagnosis is still sitting in that block of tissue just because it's too manual.
Right.
That's a reasonably high chance. So that's one reason that this manual process is suboptimal. Another big reason is that we lose the basic understanding that biology takes place in 3 dimensions. The way that we conceptualize, like even our basic researchers, when you're looking at pieces of tissue, we are taking these 2-dimensional samplings and we are extrapolating out what we think the rest of it looks like. And we have very little understanding of the, how these things relate to each other in 3D space.
Right. It's like, I guess an analogy would be, imagine if we had to debug how an airplane worked by taking, a 1-inch cross section from it.
Exactly.
Right. I think that's like a more physical analogy that hopefully our listeners can, picture in their head. So you're almost like automating all the slices. So your system will just do a bunch of different slices a lot quicker and a lot cheaper because it's not manual.
Yeah.
Let you build like a tomography, a 3D sort of graph of what all the cross sections look like.
Yeah. So we built a robot that takes the place of a lot of that human work. And so what we do is we take a diamond knife, which is the same kind of knife you would use on a microtome if you're doing electron microscopy. And we shine light through the back of the knife, so the knife becomes part of the optical train of the microscope. So as we're taking a section across this paraffin block, the tissue is sliding up onto that knife, light is transmitting through the knife, through the tissue, into optics that are focused right on the bevel of the knife, with a line scan camera in the back. So as soon as we've taken this section, we've also taken a scanned image of it. This lets us automatically cut through 10,000 sections where, a standard histologist would take this representative 5 to 20 sections. Right. This gives you much more data, much better understanding of, looking for these kind of cells in a haystack. But it also lets you take that stack of images, and because they're perfectly lined up, because we did this by a robot that's on an automated stage, we can turn that stack of
like a 3D model
into a 3D model.
Are you still doing the biopsy to get the source cells for that?
Yeah, you have to pull it out of the body first, and then it's in the mold, I'm
Yeah.
just curious from a timing perspective. Does it take, I guess, like for a manual person, 15, 20 slices is like a couple hours of work?
15, 20 slices is for someone who's highly skilled, like half an hour. For someone who's sort of a newer and less good at it, about an hour.
And then this 3 scan technology, that's 10,000 slices?
Yeah, so our 10,000 slices is going to take more like 8 hours. So it's a longer process. And a lot of what we're doing right now is focusing on preclinical drug discovery. So we're using these highly accurate 3D models. So a really good way to think about our models is to think about an X-ray and a CT. So an X-ray, CT is a stack of X-rays, but lets you see the 3D structure underneath. The 3scan is really similar. You're having a stack of 2D images that let you see the three-dimensional structure underneath. So we're using that to look at preclinical drug discovery, primarily cancer therapeutics, and let you see how blood flow and other factors are changing in between, adding your therapeutic and not adding it. So making mouse models more accurate.
So, one question I was gonna say is when you're taking the biopsy, is there also a sampling issue, like where depending on where you're taking those from?
Yeah, absolutely. And this is something that we've put a lot of work and science into minimizing, but there's absolutely a sampling issue. If you take that biopsy from the wrong place, you're not going to get the cells that are going to give you.
Like no matter how well you analyze the biopsy, 3scan, like you're.
Oh yeah, I mean, if you've taken, you're taking a sampling of a sampling, right? So there's always going to be a chance.
If your treasure map is wrong and you're digging in the wrong place,
Yeah.
there's no gold there. Yeah. No matter how good your shovels are.
Yeah.
Absolutely.
Yeah, exactly.
Any other thoughts? Or is it something that you're working on too there, or like what's the state of the art with that?
With samplings of samplings.
Make sure the sampling, the first target is
Yeah,
right.
I mean, it changes depending on what disease model you're looking at. Right, so a lot of diseases have changed their the treatment outcomes. So like something like prostate cancer, it used to be that if you had high PSA levels, your doctor's going to go in, they're going to do a biopsy, they're going to look to to see if there's cancerous cells in your prostate, and if they find that cancer, they would remove your prostate. The treatment is now changing to say, hey, actually, most prostate cancers aren't killing people, and the side effects of not having a prostate are pretty shitty. So let's just monitor it and make sure it's okay, and take more. We'll just take a biopsy, or we'll take like 12 needle cores,
right.
Every 6 months, right? So that, it changes depending on the disease and what you're looking for. Yeah.
And this and your technology is in practice, right? I recently read that it's deployed at a few different research universities. This is not vaporware. This is in practice.
This is real. And you guys, if you're in the mission, you should come by the lab.
Okay.
I'll give you a tour.
Cool.
Yeah, it's pretty neat stuff, especially when you see it. But yeah, our technology is totally in practice. Like I said, what we're doing with it right now is we're really focused on preclinical drug discovery. Being able to make these highly accurate models of a tumor in a mouse, looking at different cancer therapeutics. We work a lot with looking at models of blood flow, so using quantitative comparisons of the vasculature of a mouse tumor, before and after therapeutic or different treatment methods. So we're really focused as a business on getting our tech into the drug discovery market,
right,
while we're building out the pathology platform. Pathology is something that is hard to break into, both because pathologists are not super into automation and are not super into change. And
you're taking their jobs essentially, right? It's like,
yeah, I mean, you're making their job more accurate, but yeah, yeah. At the end of the day, you're taking their job. Though something like radiology uses computer-aided diagnostics all the time,
right,
whereas pathology doesn't. But the other thing is, anytime you're dealing with humans and diagnosing humans, you're dealing with the FDA, and you need to make sure that your technology is really robust. And so we're using preclinical drug discovery as the way to get our technology out there, and really,
doing it right,
utilize it. While we're building out the platform for pathology.
Yeah, no, I think it's really interesting. I think that the story arc of a software company or app company is really well understood. But for 3Scan, this is a mission that's been going on for 5-6 years now.
Yeah.
How did you get started? I think it's there's a lot of blogs around. Hey, I started computer programming when I was 18 and I built my first app.
Yeah.
How does one get into building out medical technology?
Yeah, I mean, it's
what's your personal journey here?
It's definitely a different world, like people in the hardware community like to say hardware is hard.
Right.
It has, longer lead times, much more capital intensive, and the same is true for biology. So having that overlap between hardware and biology is maybe not the easiest path that one could choose. My co-founder, we have a founding team of 4 of us, and so my original co-founder, Todd Huffman, worked on the core of what has become our technology while he was in grad school. And at the time, it was being used for a really narrow neuroscience application to do neuronal tracing and connectomics. So thinking about the brain like a circuit. One of the problems with connectomics is you can't see where the neurons are going using traditional imaging methods. If you're using something like CT, you're not able to see something as fine as a single neuron. But if you're taking sections by hand, you're not able to trace where that neuron goes in 3-dimensional space. And so he and I, were friends when he was in grad school, and we both thought this technology was pretty neat. But the computational overload was too much. Like, one of our cubic centimeter data sets in black and white is 2 terabytes. And so in 2008, that's a non-starter, right? But as computing got cheaper, we really saw this as being the future of medicine, the future of drug discovery, the future of biology is just moving in a more quantitative direction. And so, he and I moved to San Francisco. We pulled in a mechanical engineer, Cody Daniel, and a data scientist, Matthew Goodman, and the 4 of us lived together and started seeing if we could, build some prototypes and get some traction.
So it's still very much a startup story where it's just like get, the original key brains in place and just grind it out, like build like the, first prototypes, very much like, a Facebook story of like kids coding in a garage or something.
Totally. Still very much your standard startup story, except with these longer lead times and, things like bizarre miscommunications. Like when I thought I was being FedExed a bladder and then 2 cadavers show up on my door.
Wow. FedEx can carry cadavers?
No, a medical examiner came in with them in a gurney. And I was like, I thought you said bladder. He was like, no, cadaver. Didn't know how much tissue you'd want. I'm like, put them over there, I guess.
Wow.
Yeah.
And I'm just curious, would you even store? I mean, these are these like preserved bodies, or literally a rotting?
No, they're preserved. They're
like we need like a vault
fixed.
to like make sure there's not decomposition in our office now.
No, they are fixed and embalmed. But yeah, you have your more traditional like Facebook startup story where people are trying to scam free Wi-Fi and we're trying to like scam free tissue off people. Like my mom lives in Mexico, and I went to visit her and went to the dermatologist because my mom was getting Botox, and I started talking about what we do, and the doctor there was like, "Oh, do you need samples?" and just started giving me like
extra
blocks
flesh.
and blocks. Yeah, she's
oh
like,
my God.
None of these people need it. So I've lots of stories like that of like having a purse full of like blocks of thyroid or digging through something on a date and pulling out
flying that back into the country.
Yeah.
So one of the big questions with biohacking enhancement and therapeutics is just longevity effects, like things that affect over a long time scale, because that's just hard to do, right? There's twin studies that when you're trying to really look at how something affects someone in the long term, that's just it's a hard problem because time has to pass.
Yeah. Absolutely.
Are there things at the tissue level? Are there techniques to accelerate time and to aid the study of things, of knowing, hey, if I do this thing for 40 years, am I going to be better in 40 years based on my genetics?
Yeah, I mean, I think there's a few different answers to that, right? Like, absolutely being able to combine these different parts of biology that we understand makes us better able to make predictions. So, one of the things that's happening in the world of genomics, right, is, we sequence the genome. This is amazing. You get these crazy, get all this data from it. No one knows what to do with the data. We spend, years analyzing the data. We start to see, correlations, and we come up with this whole field of bioinformatics to help us understand the data. Now, and we're as a scientific community, super jazzed, and we've got this blueprint to people now, right? And then now we start to see that the way that we looked at taking these samples, we would take a piece of tissue, we'd essentially throw it in a blender, and then sequence a part of it, right? Now we find that sequencing one part of it versus a different part gives you a totally different answer, right? Being able to take the learnings that we're getting from genomics and put them into something like what we're doing, where you have a spatial mapping of tissue, and also using techniques like next-gen sequencing, being able to sequence something off of a single cell, right? But then overlaying that back into our understanding of structural biology, and taking our understanding of structural biology and being able to make that one level up, and then see how that fits into the context of something like an MRI and like an in vivo study. Like, all those things need to be correlated, and the correlation between those will definitely give us much more information than what we have now. Nothing speeds up time, though. Like, you still aren't going to be able to get 9 women to make you a baby in a month, right?
Well, so then a related question is then, like, how close are we to completeness as far as just having an accurate computer model of a human body that's just this is everything, from structural to tissue to nano, like every sense of a human body. This is how it works.
Yeah.
Like, and then we're done. Or like if we get reasonably close there, then you could accelerate time on that model. That's really interesting.
So I think that we're much further off from that than people realize. This same project.
I think the base science that we don't even understand how everything works. First of all, right?
Yeah. I was actually really shocked.
The tools aren't even there to just resolve how everything is put together.
Yeah, totally. I mean, I was really shocked with that project I was talking about, where I was expecting to get human bladders, and instead I got bodies. That project was to map the innervation of a human bladder to make a more effective overactive bladder treatment. They're trying to kill some of the nerves so that you wouldn't have the same bladder input. When I was talking to these researchers, I'm like, well, what are you using to map it now? Like, I have looked at anatomy textbooks, like I can see nerves in there. It's like artist interpretation. We have no idea where the nerves are. None at all. So even something as large as where are the nerves in your body, and these are researchers who are in phase 3 clinical trials of their treatment. Like, they didn't know where the nerves were, because that's something that is basically impossible to do by hand.
Wow.
Like those are still gaps that we have. So we're still a fair ways off, but the more we're able to do this cross-modality stuff, right? The more we're able to get the genomic into the tissue, and the tissue into the in vivo, the closer that we get.
Yeah, I mean, I think theoretically there's no computational limit that we can't model this out. But I agree. I think the state of the art is that like even just like resolving how like neurons are connected to each other, right? Like that's just hard to do, right? And I think when people are looking at all those like philosophical questions, can you even like map, if we're to clone your exact like state into another person, like how does that even work, right? And like there's a lot of questions around, even if you're imaging things, like that would like just by observing like your like the I guess the neuron connections or the quantum state there, right? Like that would change your brain state. So like basically like it's hard to even like find like base truth of how you're
Yeah, absolutely. I mean, by imaging it, you're definitely changing it. And, some of those basic questions, like when the lab that the basis of our technology came out of is this brain networking lab, and, looking at connectomics, what they wanted to do was be able to get good imaging of neurons and how neurons connected to each other so they could run simulations of it. And even that is, it's hard to get the degree of granularity. Also, more and more in biology, the more you learn about something, the more you know that you don't know. I mean, when we sequenced the genome, we thought we had this,
map,
the blueprint, we had the map,
Right.
right? And there wasn't any conversation about epigenetics and how, different, you're going to see different things at the tissue scale in different environmental conditions.
Right.
So it just, I think, really shows the more that we need to push on it to be able to see all of the things that we currently don't know. I also think it's one of the reasons that biohacking is really interesting and really important. I think it's really important that we don't, that we feel comfortable to change our own bodies in the way that we want to change them. And one of the things that I think is really awesome about what you guys are doing is making, nootropics and, like, biohacking something that is like real and not fringe and accessible to people.
Yeah, and tangible. Yeah, exactly.
So like I have a magnet in my finger, right? And like one of the things that I think is really awesome about it, right, is I think that sense of comfort of altering my own body to do the things that I want it to do is incredibly essential. And that a lot of that is just, like a psychological thing, right? Is like feeling comfortable with doing it. And like, we're not going to know. We're not going to be able to run the simulation to know what things are going to be like in 40 years. So you just need to feel okay to take control of your body, and whether that's through nootropics, whether that's through implants, like we should feel like our bodies are things that we can hack and we can change.
Yeah, I think that's how progress is made, right? I think if you have like a theoretical understanding and some early data points, that's how extrapolation happens, right? Like that's how Wright brothers were like, hey, we can actually fly something.
Yeah.
Like you got to experiment and tinker with it to see what is even possible. Yeah, no, I think that's a great segue actually. I mean, thanks for even just bringing it to biohacking. Obviously as we're building a biotechnology sort of microscopy company, you're looking at all these different ideas. What got you into experimenting, with different biohacks? What got you to implant a magnet into your finger?
Yeah, I mean, I do just think, that I think that there's so much that is biohacking that people have normalized and just don't even consider it anymore. Like we use biohacking just to refer to the things that we see as being fringe. But like I was putting on makeup this morning, and that's biohacking, right? Like I'm actively changing the way
your
that
appearance,
that people are seeing me, to trigger some, evolutionary signals to you that I am, younger or have different facial bone structure or whatever, right?
Right.
And we don't conceptualize that as biohacking because it's completely normalized. So anything, I feel really strongly that by having more people do the things that are on the fringe, we make those things on the fringe more and more,
Normalized.
okay. Yeah.
I think it's a great point. I mean, we talk about like eyeglasses. I mean, that's like one of the another original biohacks, right? Like it's not natural. It's not a God-given thing to have like, things on our face make us see better.
Yeah.
But now if you're not wearing glasses and you have, nearsightedness, you're like a broken. It's not a zero-sum game to make progress. I think progress is inevitable if you just look at what makes us different from other animals. It's by being able to pass along technology, by passing along advancements and how we can manipulate our environments and ourselves.
Yeah, absolutely. And of course it's complicit. Like living in a house is complicit,
Right.
Like, we are not able to be out in the elements, and those, and if we try to, like if I move to Alaska and don't build myself a structure, like of course I'm going to die. Yeah. That, any form of modification like makes everybody have to do it. That's what being evolutionarily fit is.
Yeah, I think it's funny because like where's the line? If it, like some seem like, oh, that's okay stuff to like be doing to our environment or ourselves, and this other stuff is like unnatural or fringe. And I think that like just as humble human beings, like just being saying that, hey, there's a magic line that I get to choose is like I think very arrogant. I think it's just like let people get educated and have them choose their own things that they want to do.
Yeah, absolutely. And we do have a culture of deferring to authority, right? Like diagnostic tests. This is a problem that 23andMe has went into, right? That like diagnostic tests have to be administered by a doctor. There's a great article that I'll send you guys about home pregnancy tests, talking about how we should never. It's an article from like whenever home pregnancy tests came out, I think like the 20s or 30s, saying, warning us against this horrible world where women could find out that they're pregnant and not understand the concept of it, and they would go off and kill themselves, and they would go off and like do these horrible things.
Right.
This idea that we need to protect the weak and delicate members of our society from their themselves is really fundamentally wrong.
Yeah, I think it's patronizing, right? Like if you're not educating them, you're keeping them continuously boxed in some cage, right? Like I think the better solution is to increase education. Be like, hey, this is the actual facts. I think it's like, right? I think that's like as entrepreneurs, as people that are adventurous, I think we've all seen that. Hey, you can learn a lot by you don't necessarily need to be taught with like in a college or in a classroom. A lot of things you can learn by reading the source material.
Yeah, and I think this is one of the things that we, as a society, with the advent of the internet, we now have this, rich trove of information that is generally accessible to everyone. Like, I am a believer that you need to have, a solid education system that is freely available to everyone.
Right.
So that you can contextualize and understand information. But we do live in a kind of magical time where it's reasonable for people to get information, understand it themselves, make their own, assessments, and decide for themselves what they want to do with their bodies.
Yeah, absolutely. I'm curious, from just a more tactical question, like how is it to have a magnet in your finger?
Oh, it's great.
Describe it for our listeners here.
It's great. It's it's like having another sense, right? So you can tell when electromagnetic fields are around. You can tell when things are on. And like from a practical perspective, other than holding bobby pins when I'm doing my hair or picking up small screws when they fall on our optical bench, like there's not that much that it comes up practically, but it's really cool to be able to have changed my own body to give myself a sense that I wouldn't have otherwise.
Do you like change out magnets? I've heard that magnets get demagnetized over time. Do you like sort of swap in different magnets, or have you had to have that issue that your magnet hasn't lost power yet?
Yeah, and I don't believe that the magnets really lose power over time. Actually, so my co-founder and best friend, it was the first person to ever do a magnet in his finger, and his has not lost any power.
Okay. Yeah. What's next for you guys? I mean, after that Series B, obviously, I imagine that you guys have a big roadmap ahead, and potentially hiring people. If there's listeners out there that are resonating with Megan's culture and the company mission. I mean, I'm curious.
Yeah, absolutely.
Have you just open up just areas that you're interested in and looking for talent?
Yeah, totally. I mean, really, we're excited by anyone who is excited about what we're doing. So if you think that this is something that really resonates with you, if you have skills that you think would be valuable, like, come to us and let's talk and see if there's something that if there's something we can find. You can go to our webpage, www.3scan.com. We've got we have a space there to contact us. We do list job opportunities for things that we're looking for, but we're also really into hiring people who have those those intangible skills that we didn't know that we needed until they come to us. And we love talking to people. We love having people by. We do monthly happy hours as a nice way to bring people by.
Okay, so lucky SF people try to stop by then.
Yeah. So really, if this is something that resonates with you, just contact us.
Cool.
Yeah.
Yeah. I mean, is there any other last words in terms of biohacking? I mean, I think we covered an interesting spectrum from your personal story as an entrepreneur, your technology behind 3Scan, and how it's going to impact and sort of change how microscopy is going to be done, towards your personal experiences, seeing that, hey, biohacking should be for everyone. It's something that we've all been doing, and it's just like this weird, I guess moniker that's been recently growing a lot of traction. Any last thoughts, concluding statements around anything you want to mention?
Yeah, I mean, really just love for you guys and what you guys are doing. I mean, I really think that by bringing like nootropics to to a mass market, and it changes the way that people think. It changes the way people thinking from it changes people from thinking that nutrition and food is this thing that you are told, to it being something that you should look at, that you should be looking at your brain chemistry. You should be looking at, what tools you need to give yourself to make yourself smarter and better. And that spirit of adaptation and change, permeates through everything else. And like that has huge, widespread, like world-changing value. And I love what you guys are doing.
Thanks for the kind words. I mean, well said. Not much. I can't really add much there.
All right, that's everything. I mean, thank you. Thanks for your time.
Totally.
I just want to say to everyone out there that check us out on SoundCloud and iTunes, Google Play, and YouTube. There we go. Yes.
For the video of you can see Megan and her blue hair. We'll have all the links that a couple of the resources that Megan mentioned as well. Catch you guys next week. Cheers.